Actually, those can block DNA RNA since it had some activity as well, along with some macrolide antibiotic has some activity also. So moving on to this further evaluation of these drug candidates, in fact, this is a study done by a master student from my lab, Hector, is doing a great job as part of his SCM thesis, where he evaluated some of the drug candidates, especially, I know there's a lot of interest, Dr. Rajita is going to talk about his findings about disulfiram and azlosilin. So Hector actually compared these. This is actually all these drugs is a stationary phase bacteria old culture treated with 50 micromolar of these drugs. Then for 1 week, then look at the survival rate. And as you can see here, these are current Lyme antibiotics, and then we see actually the azlosilin and disulfiram have similar activity, and they don't really have a very strong anti-persistent activity, certainly not as strong as the ones we evaluate, like the daptomycin, clofazimine, even clarithromycin. The other one we found quite active is cryptolipine. It's the active component of cryptolepis, which is a natural product, which I'll come back to. So if you look at the activity of these drugs against the growing form in terms of the MIC, against the growing form, then these two drugs have good activity there. For example, azlosilin is more active than amoxicillin, but is not really as active as ceftriaxone or even clarithromycin. Disulfiram has activity against the growing form. Yeah, it's 0.3 to 0.6 microgram per ml. Yeah. So according to our study, it looks like these two drugs have good activity against growing form Borrelia, but not so good. Had some activity, I would say, not so good as against the persistent forms. So this is actually the microscopy data that corresponds to these percentages, where you can see the green, the control untreated, a lot of them are live, 92% live cells. Doxycycline, you get a lower percentage, 67% live cells. But then you have some other, daptomycin is very good, 0.2% viable cells. Clarithromycin, disulfiram is the data I showed you. You still have some green cells remaining. That has some activity, better activity than doxycycline looks like, but not really as good as some of the others we evaluated. So we found that even though we have the persistent drug, we can't use them alone. They have to be used in the context of drug combination, as I showed you in the yin-yang model. So we need, or in fact, we found the most effective one is a 3-drug combination. So ones we found is the daptomycin, doxycycline, cefuroxime. But this was all published, so I'm not going to go into detail except to say that even though this is powerful, daptomycin is a very expensive IV antibiotic. We're interested in develop to replace daptomycin with oral persistent drugs that are equally or perhaps more effective than this combination to kill the persistent forms. So this is actually just some additional slide to show you the combination of some of the drugs we evaluated that show promise. You can see red, that's what you like to see, because the red indicates the bacteria are dead. This is our control, so this is actually when you combine the two current Lyme antibiotics together, this is untreated, the treated, that is a 40% still viable, but this is our triple drug combination, the daptomycin drug combination we use as a control. And these are the 3 examples we showed that have good activity in combination. Is clarithromycin plus nitroxyline, as well as linezolid, and this is a 3-drug combination. So this is the slide we published earlier, where we also evaluated different drug combinations, including HCQ. You all probably know about this. Is a hydroxychloroquine, which is really the drug used for treating COVID-19 has showed some promise there. And it's interesting, it's also used for treating Lyme arthritis. So some of the Lyme patients may take HCQ, hydroxychloroquine, that may actually protect them against the COVID-19. So I thought it would be of interest in such a current environment to mention this potential interesting observation. So then we went on to to look at some other treatment possibilities, looking at the natural products like essential oils. So we've done 2 studies on essential oils. First one, we identified 3 most active ones like oregano oil, cinnamon bark, clove bud have a very good activity. Then in the second study, we identified another 10 of them look very good. The garlic oil, allspice, myrrh, Hadecam, Lysicubiba, etc. And we showed that, in fact, this essential oils in combination with the current Lyme antibiotics has shown good activity, like all these. So I'm not going to go into detail. So this is, by the way, I want to just tell you that this is all in vitro study so far, okay? And I'm going to tell you about the animal study later. So also evaluated this natural product and the herbal medicines. These was done in collaboration with Dr. Sanjay Schweig from California, as well as Leon Jacob. Jacob Leon. So where we evaluated a range of herbal medicines that are used by Lyme patients. This is largely derived from the Buhner book, this Lyme herbal medicine book, where we evaluate a panel of about 14 natural products. It's interesting. And we found actually 5 of them to have very good activity against Borrelia persister forms, as well as against the growing form. So the one, for example, Cryptolepis, okay, is very potent, very active. The other one is the Japanese knotweed, which is an invasive species in the UK. They try to get rid of it. So it's a very good plant that has good anti-Borrelia activity. Then the other one that's pretty potent active is black walnut. Cat's claw as well is very good. Artemisia annua, the herb, is also has some activity, but not as strong as the ones I highlighted. So the ones I highlighted in yellow turn out to have activity against Bartonella as well. So this is a study which not yet published. So this is the study, yeah, now that I'm talking about Bartonella, we also did FDA drug library screen against Bartonella because it's an important co-infection pathogen. So these are actually currently used antibiotics for treating Bartonella infections are listed in black here. The aminoglycoside, this macrolide antibiotic, rifampin, doxycycline, etc. Then we found those highlighted in red to be more potent, more active than the current existing Bartonella antibiotics. These include daptomycin again. So we get this one drug that has activity against both Borrelia persister and the Bartonella persisters. Now the interesting one is methylene blue. It's actually very potent, very active against Bartonella persisters. The antifungal azole drugs, nitroxyline, it's an antibiotic for treating UTI, has some good activity as well. So in drug exposure studies, you can see those are the really kill completely the bacteria in terms of colony count. So what we do is that we expose an old stationary phase Bartonella culture with all these different drugs at 50 micromolar. Then at different time points, 1 day, 3 day, look at the number of bacteria that survive. So you can see those highlighted in pink are the ones that are most potent: methylene blue, daptomycin, azole drug, nitroxyline. So among the existing antibiotics for treating Bartonella infection, the ones aminoglycoside, amikacin, kanamycin, pretty good. By day 3, kill all the bacteria. And the other one that's active, second to aminoglycoside, is rifampin. Okay, 10 to the 3. So all the others pretty much similar. So this is all in vitro. Then we also went on to evaluate the essential oil panel on Bartonella, where we found again some of the overlapping essential oils have activity against Bartonella stationary phase form as well, like oregano, cinnamon bark, clove bud, allspice. All this highlighted in red are the ones. Now I'm going to move on to the animal studies, where we have two types of animal models. One is this long-term called type 2 persistence, which is long-term persistence model, which is a study established, the model established by Amir Hodzik and Steve Bathold at UC Davis. This is a study we're doing, evaluating different drug combinations with our collaborator, Monica Embers at Tulane. So got some data there. So I'm going to talk very briefly about this because this is not published. Then our published work on this using this type 1 persistence model, starting with a persistor inocula, then develop a more rapid persistence model, which is a Lyme arthritis model. So with different inocula, so this is where it's important. I think previously people's attention focused on this log phase, spirochete form of the bacteria, but we believe that may not be the most important form. And then in the older culture, they developed this round body form as well as the microcolony aggregated form. These are more tolerant to persistence. So the current Lyme antibiotics don't kill them very easily. And these, not only that, not only tolerant to antibiotics, they also cause more severe disease. So it's actually shown in the mouse model here. This was published earlier last year, March 2019, where we evaluate these different forms, the log phase, stationary phase, as well as microcolony form, injected into mice. Then at day 21, you can see the microcolony form cause a more severe arthritis, a swollen, more swelling in the joint. But the log phase is not yet able to cause very severe arthritis on day 21. But on day 35, the difference between these two groups getting smaller because this log phase, stationary phase, that caught up. They actually develop a more severe arthritis, but still the microcolony-induced arthritis still remain more severe, although even though it can go down in severity as time goes on. So this is actually also shown by histopathology, heart base, as well as joints. So I'm not going into detail here. I'm going to move on. So based on this mouse study, we propose that in fact there's two types of a persistent Lyme disease alluded to earlier in my presentation. So this is a type 1 persistence, this is an early development persistent Lyme disease. This is essentially PDLDS. That is, this is a type of persistent infection that's introduced very early during the tick bite. So depending on the Borrelia bacteria in the ticks, so the hypothesis is that the Borrelia bacteria residing in the ticks are very heterogeneous. So it's depending on the particular Lyme bacteria being introduced into the body that can determine the disease outcome to some extent. So if it's a log phase growing form of the bacteria, that can be easily killed by the current Lyme antibiotics. So the patients are cured. So that's why you get the 80 to 90% of the patient population cured the current acute form of the disease. But in some cases, people may get unlucky because the tick may carry this biofilm or persistent form of the bacteria that's introduced. Even though you treat with a single antibiotic, you don't cure it. We know biofilm infection cannot be cured with a single antibiotic. So then you have the late development persistent Lyme disease. That is, the initial infection was not diagnosed early enough, treated on time, then developing into this persistent form of the disease. So using this type 1 persistence mouse model, we evaluated a different mouse model. We developed this log phase, the spirochete form introduced infection, as well as this microcolony, the persistent form, introduced infection. Then evaluated different drugs, different drug combinations. Then we looked at culture as well as PCR of the tissues. So by culture, we can see the microcolony introduced form produce a more persistent infection that's not easily eradicated. Of course, majority of them are. You can see there's a number of mice that are positive still remaining. So microcolony introduced form of infection, ceftriaxone treatment still has one mouse remain positive by culture, but then by in the log phase form introduced infection that's easily cured. Then we show that in fact the daptomycin drug combination is able to eradicate the infection by culture as well as by PCR. So the PCR data is shown in brackets in here. So this actually validates our in vitro finding in the mouse model. So here I want to really mention this very important concept of these different levels of persistence. So you can produce a mouse model that is easily cured with a current Lyme antibiotic, as well as you can establish a more persistent infection that the current treatment, like even ceftriaxone, the most powerful antibiotic, cannot eradicate. So it all depends on the model. So now moving towards the end, in fact, so for the future treatment of Lyme disease, so in the short term, I think we can use this, still use this IDSA treatment, but then we'll have 10 to 20% patients not cured. So these are the candidates for testing the persistent drug regimens we identified. But in the long run, once we identify some of the optimal persistent drug combination, they can potentially use to start from the very beginning so that we don't even get the 10 to 20% patient not cured. So we can get a much better cure for the disease. So to move forward then, so I think even though we identify some promising drug combinations, I want to emphasize this is a long-term thing, and we still need to evaluate all these different drug combinations, especially the combination essential oils, because there's a potential side effects of the chemical drugs, then some natural products as well as essential oils look promising. These need to be examined as well in terms of their active components, their utility in vivo in the mouse model. Now that we identify some promising drug candidates, I think it's important to move on to clinical trials to evaluate these persistent drug combinations. And it's important to determine, try these on appropriate patient population, as well as we have to know really how long to treat. We can't just treat for 30 days because the 30-day is what we treat for active disease. For the persistent form of the disease, you need to treat longer. But the optimal length of the treatment, I think we need to determine clinically by the proper designed clinical trials. And then we need to identify biomarkers for treatment response, as well as address the co-infection problem and the immune suppression. And we can't do all this without funding. And finally, I really want to end my presentation with this yin-yang model, which is really try to bring some unity to this field. That is to propose this yin-yang model to try to unify the field, where the yang means the active form of the disease. That's the current treatment helps. But then we have the yin, the persistent form of the disease. Then we need a different approach, which is a persistent drug combination as shown here. So this yin-yang model actually also shows, not only sort of tells you about the two forms of the disease, this active form of the disease that's really taken care of by the IDSA approach. Then there's also the persistent form of the disease that's really largely pushed to this ILADS physicians, LLMDs, who try to take care of these PDLDS patients. But then this model also provides a solution to this problem. That is, we need a persistent drug combination which we found some we think have potential to really bridge the gap and to really solve the problem facing the field. Try to solve that. I mean, as we all know, this is really a war. It's really partly because the current science is not quite there yet. We don't quite know the persistent infection very well. We're only focused on the small acute form of the infection. This is not only unique to Lyme, in fact, many other persistent infections. In fact, this is where it leads to chronic form of the disease of unknown etiology, autoimmune diseases, diseases of unknown etiology, like CFS, chronic fatigue syndrome, MS, Parkinson's, you name it. So I think, I often get asked the question, where do I stand in this debate? I can tell you, I stand on both sides. I stay here, right here in the green dot. So I think with this yin-yang model, we'll be able to unify. I think the more important thing is that if we are able to really show the efficacy of the persistent drug regimen, then that would actually help to resolve the controversy and help patients, I think, above all. So finally, I want to thank the people who have done the work from my lab, GFN, and many others who have done